So we're going to do a quick review with membrane transport and then figure out where we're going with cellular respiration, just based on some of the formative assessments we took today. I think there's some key points that we have to make sure everyone understands. Number one, when we're talking about membrane transport, remember, this is important to remember, and I thought it was clear. Ionic, right? All right, or charged particles, right? Charged particles do not pass through the membrane without help. That's it. I mean, there's no ifs, ands, or buts there. If an ionic compound or an ion, a salt, an Na plus, okay, or Cl minus, or a potassium plus, or anything that's charged is trying to pass through that membrane, it needs help. And what kind of help can it possibly get? Well, if you have these phospholipids, and remember they're phospholipids, so the lipids part is the problem is the part that has a problem with the ion. Then you need to have some kind of protein, some kind of integral membrane protein that's going to help that ion through the membrane. That protein can come in different forms, and we'll talk about the forms again in just a second. But for sure, the ion there has to be some kind of opening, right? And remember, we talked about this, that inside this opening has to be polar or hydrophilic, right? So that things like the sodium, like the chloride, like the potassium, can flow through. The outside, this part outside, let me change colors really quickly. The outside, it has to be hydrophobic in order to be able to sit inside this lipid bilayer. Otherwise, these lipids would kick it out. So when we're talking about this charged particle business, these charged particles, they will flow through this protein, and that the fact that if it flows through these flow through the protein, this is called facilitated diffusion, right? So we call this example facilitated diffusion. And facilitated diffusion, oh, look at this. So it's called facilitated diffusion. Facilitated diffusion. So we're still getting diffusion here, okay? And why is it diffusion? It's diffusion because the ions are going from high to low. That's the diffusion part. And it's facilitated because the word facilitated means help, and it's being helped by this protein. So it's facilitated diffusion. And that's what the video that I sent you a couple days ago kind of goes over in, I think, a pretty good graphic way. So this is facilitated diffusion. This is the kind of thing that happens in the mitochondria when we're talking about the hydrogen ions, but I want to use that as a final example. But this is facilitated diffusion. This is a diffusion or movement of ions through the membrane. It needs help. If you don't see a protein in the membrane and they talk about the cell membrane, or they talk about this membrane and they don't draw a protein, then it's not going through the membrane. You need to have that protein there to help these through, because this nonpolar lipid layer won't let it through. I hope that's clear now. I can see where some of you might have messed up after I thought about it a little more and considered the example we had in class of the mitochondria and the question that the state asked on their practice exam. I can see how it was a little ambiguous for people. But what the state was actually trying to figure out is, did you know, are you sure you understand that you need a protein for this stuff to get through? And they drew you a membrane that didn't have any proteins. And I don't think a lot of you noticed that. So they kind of tricked you, but they didn't. They were just testing to see if you understand that a protein needs to be in this membrane. So be careful. They're going to test you in a number of different ways. You could understand most of it, but one piece is missing and you didn't think about it, man, they could slam you, and that's what happened today. All right, let's look at the next question, or let's look at the next piece here. So we talked about ions. What other things can't pass through the membrane? Anything that's big, any big molecules that are charged. Okay, so charge is no good. All right, so things like proteins. Usually, most proteins are not going to go through the membrane. Things like sugars, right, are not going to go through the membrane. And you guys should know that, right? So big, they're charged, proteins, sugars, that's a no-go. No passage. Not without help. We can get it through, but you need help to do it. All right? So what does get through? What does get through? All right, what does get through the membrane? And what does get through is they're small, obviously. So everything that's opposite what we just saw. So not big, but small. Not charged, but not charged. So it can't be charged. So they have to be neutral. And usually, as we said earlier on, which is why we talked about it for so long, not charged, we're talking about gases, right? Nonpolar molecules. We're talking about gases here, people. And so gases are nonpolar. They'll pass through without a problem. Okay, they'll go through the membrane, no problem at all. And lipids, small lipids, right? So small, but lipids as well. Lipids will go through easily enough. So lipid soluble stuff will go through the membrane. That's not a problem, because lipid soluble, the hydrophobic side of the membrane, the lipid layer isn't bothered by lipids coming in, because like dissolves like, right? So the small lipids, the not charged gases, those will fall through fine. But big, if there's charged, if there's proteins, if there's sugar, they're not getting through. All right, so how do we get these in and out? And we've covered this, but let's review it really quickly. We talked about facilitated diffusion, right? So you have the membrane here, and then we have this protein that sits in the middle, and they'll be drawn in different ways, okay? So don't get hung up in how I draw it. They could be round, they could be whatever. There's this tunnel here inside the protein that allows things to go in and out. We could have that. It could be, another way we could do it is with, we could have a co-transporter. Well, that would be a set of active transport, but this is passive transport. So we're going to talk about things coming through quickly, diffusing through, using these proteins, and that's pretty much the only way it gets in if they're small. Now, there is one more thing though, and that's what happens when things are too big to go through these small openings, right? What happens when we're talking about something that's really big? Okay, when we're talking about something that's really big, then what the cell does is it changes, the membrane swallows it up. Oh gosh, I'm not sure how to describe it, but to say watch the video and look at it and see how it's done is that the membrane actually starts to change and bend and fold. And so what you have is, if you have this cell and it's trying to eat this big old piece of or maybe a large amount of ions all at once, or a larger piece that doesn't fit through an opening, then the membranes actually, as this protein gets closer and closer to the cell, all right, the cell starts to change. The membrane starts to dip a little bit, all right, and it's still going to be round, and you're still going to get this. You're still going to get that little. The thing's going to be in there, all right, and you're going to have the little. It's still coming to get surrounded. But slowly but surely the little piece is surrounded by the membrane. And what happens ultimately is you have a cell, as the membrane kind of breaks off, pinches off, is inside you have a membrane, and your protein is inside. And at this point, that's when a vacuole comes along and fuses with this and digests it, and then all that gets released into the cell, and then all the pieces get taken in and distributed to all the places in the cell needs to go. So this process that we just outlined, and as we discussed before, is called endocytosis. And it is a really it's just we don't really call it active diffusion, active transport. It is active. It needs ATP. This is energy. We need definitely big energy here to do this. This is passive over here, right? So passive diffusion or facilitated diffusion, where you have a protein that's helping it. These are passive, no energy, all right? No energy means no ATP. Okay, when I say there's no energy required, no extra energy required, that means no ATP. Or sugar, no sugar required, okay? So I saw that a lot of people made that mistake. They sound a lot of, I don't know why, but you know, it's new, so I guess it's easy to get confused. But no energy means no ATP. So a lot of people put no energy, but then they put uses ATP. Well, that's not true, because no energy means no ATP and no sugar. That's key here. So this is endocytosis, and this is passive diffusion. Now let's talk about something else that this cell has to do. So we talked about diffusion, and diffusion is happening across a membrane. Diffusion happens across a membrane when the particles are small and not charged. And the key example would be water and gases. So water and gases definitely pass easily through the membrane. So when we're talking about this membrane here, right? And as I said in those study blue cards, I think I made it pretty clear, but I guess not, is that these little particles of oxygen gas, and there's two of them, right? Because it's O2, right? Or any CO2, right? CO2. So anything that has a big oxygen with two big oxygens with a carbon attached to it. Right, so CO2 is, these are going to pass through the membrane easily. So these two pass by easily through the membrane. Okay? So as we sit here and we look at these molecules of oxygen gas and carbon dioxide, the carbon dioxide is easily going to flow through from high to low, and oxygen will flow through from high to low. And it's not going to have any issues with that, because these are gases, they're nonpolar, and they easily diffuse in and out of the membrane. No issues. They're small and they're nonpolar. Nobody has a problem with those. So when we're looking at those are and of course water. Waters are our favorite child, that also passes through without any help. But the cell needs water to pass through so much that it actually, sorry to go real quick. Water passes in and out. It needs it so much that actually the water molecule, the cell, does a facilitated diffusion for water as well. So it has its protein, it has its opening that's going to be hydrophilic. So water can come in and out of here without too much trouble. Because water, unfortunately, fortunately, is so necessary that it's able to, the water molecule goes in and out of the cell freely, but the cell needs it so much that it makes this protein available to it to use as well. Very important molecule, obviously, and because it's so important, the cell lets it go. Of course, it diffuses through the membrane because it's small and it's polar, but it's not charged. So water can go through the membrane and it can diffuse up and left, but it can't do it as quickly enough for the cell. So the cell has these pores all throughout its membranes, allowing water to go rush in and out as it feels it needs to, equilibrating the concentration, keeping the concentration equal so that there's no issues in the cell function. So when we're looking at this, at these molecules that pass through easily, I hope no one for the rest of your lives ever say that water does not pass through the membrane easily, unless, of course, you're told in the problem it doesn't. You never know what kind of tricky question you might be asked. But water will pass through the membrane easily, always. And so will small molecules like oxygen and carbon dioxide that are nonpolar. So not charged. Water's not charged. It's polar, has partial charges, but it's not charged. They're small. They're not charged. And, of course, gases are examples of these as well. These pass through without any help necessary. They're always going from high to low. And because they're always going to high to low, from high to low, right? They do not need any energy. No energy necessary. All right? So there's no energy necessary for these to go from high to low, ever. So try to remember that as we move forward, that when we're talking about passive transport, it's no energy because you're moving from high to low. And high to low does not require any energy. Okay, what about this thing called co-transport that you might have seen on the video that I sent you? Well, now we're going to start talking about active transport. And active transport is important for the cell, obviously. And the deal with active transport is that it uses energy, does use energy. This is where we use energy. We use energy here. It's probably the biggest use of energy in most plants, in most cells. So we use energy here. Okay. And when do you see this happening? When do you need energy? When you're going from low to high. As I said in class, as I said in every example we've looked at over the last week, when you're going from low to high concentration, that's where you need energy. In the form of energy, you need ATP. You need to use ATP. ATP has to be used because that's the energy currency of the cell, and that's where you go from low to high. So anytime you have a membrane, okay, and you have a substance that you need to go from low to high, for whatever reason you need it to go. If you have sodium potassium, let's take a look at sodium and potassium, or hydrogen ions. Those are two great examples we'll look at in the next slide. Those are places when you have just a few of them, just a couple on this side, and you have tons of them on this side. That's where you have to decide as a cell, what is it that I need to do? What is it that I have to move? If it's going to be to let it go from high to low, then you're going to have a passive transport helper to help these ions just flow from high to low, because you're producing all these high atoms or ions or charged particles. If for some reason you want to move these few into the place that already has a lot, so remember this is high and this is low. If you want to move them in that direction so that you have some kind of flow that's against what it's natural, against the low energy state. So if you want to take these low and move them towards the high, then you're going to need energy, and you're going to need that energy in the form of ATP. Okay, and when do you need to do that? Well, you need to do that when you have, well, we'll talk about it, we'll look at it in a minute. But you need to do it with sodium and potassium, and then you need to do it with hydrogen ions. And let's look at these two examples that, again, we've covered in class, but I want to just quickly summarize. So let's take a look at the sodium-potassium pump. And this is a really prime example of active transport. It's the one that they're probably going to use on the test if they use it at the end of course exam. And it's used in textbooks all over the place. We've looked at it ourselves. But let's look at what's going on here. Here you have an extracellular space. So this is outside the cell, extracellular, outside the cell, intracellular, that's inside the cell. So here's ATP, 3 phosphates drawn in kind of little arrows, purple arrows, and then you have a blue triangle that is the adenosine. Here you have sodium ions, and sodium ions are out here, and you have to see the potassium is somewhere. Here it is. Potassium is this yellow or orange oval, right? I don't like that they use a hexagon because we always say it's sugar, but in this case, the hexagon is sodium. You notice the sodium is high outside. See the little graphic here? Sodium is high outside, and potassium is high inside. So again, sodium is also high outside, but potassium is low outside, and sodium's low inside. So if things were going to go normally, what would happen is what? Without energy, what would happen? Without our friend ATP, what would happen here? Okay, I would hope you'd say that sodium would move from the outside to the inside, and potassium would move from the inside to the outside. But the cell doesn't want that to happen. So what the cell does is it uses this protein that acts as a pump. So it actually literally binds then the sodium here while it's open inside, and it binds it. So you kind of stick to it, the hydrogen bonding or some other intramolecular force. They bond, and then in comes an ATP. And as the phosphate is released and now the phosphate is again, this protein is phosphorylated, ADP leaves. Notice there's 2 phosphates, so adenosine diphosphate leaves. And the protein changes conformation, letting then the sodium leave. Everybody's happy because now sodium is getting pumped out of the cell. For reasons we're not going to discuss, the sodium wants to leave, the cell wants the sodium out of its internal space. Now on the outside, the cell does want some potassium to come in. Here's potassium. As this is opened up to the outside, the potassium binds to the inside of the protein. And as it binds to the inside of the protein, it changes the protein's conformation, the shape that we discussed so many times. And when that protein's shape is changed, it releases the phosphate. The phosphate's released. And when it releases the phosphate, the potassium goes out with it. And that's how the pump works. First, the sodium binds, allowing an ATP to come in and hit this with some energy that allows the protein to change its shape. Now it's open outside. Now the sodium just is released, and out it goes into the outside. And now this is open, allows the potassium to bind. When the potassium binds, the phosphate, it changes shape again, and now the phosphorus and the potassium are released inside. Of course, this is going to be taken up by glycolysis, and it'll take up and other and the TCA cycle or Krebs cycle, and it'll be taken up by electron transport chain. Those processes will make more ATP, right? But the point is that this movement of moving these ions, the sodium and the potassium, against their concentration gradients. So now this potassium is moving in, so it's moving from low to high. So the potassium is moving from low to high. It needs energy, and that energy comes in the form of ATP. Then this sodium is going to move from low to high. And so see low sodium to high sodium, so low inside, high outside. So the sodium is moving out. And that's, again, because it's moving from low to high, it needs energy, and it comes again from the form of ATP. Never do you see sugar coming in here and giving energy directly. We don't use sugar directly. There's a whole process for getting the energy out of sugar. We have to take the energy out of sugar by using a series of chemical reactions, and those series of chemical reactions are what we call glycolysis, the Krebs cycle, the electron transport chain, or in the case of no oxygen being present, fermentation, all of which allow us to squeeze some energy out of sugar in the form of ATP. We cannot use sugar as a source of energy itself. All right. So what's the take home? What's the point? Everybody keeps telling me what's the point. I hope you get that active transport. Oh, let me change the font to the pen. Active transport is for moving things from low to high. All right. That's number one. Number two, ATP is what's used here to do this. And the sodium potassium pump takes sodium in the sodium potassium pump. It's what you're doing is you're taking sodium out and potassium in. But what's key here is against the concentration gradient. So again, what does that mean? From low to high, not from high to low. So you need that energy, and you need that energy in the form of ATP. So here is our electron transport chain that we went through in the other video that I sent you, and we went through in class, and you have your book and you're answering questions on it now. So there's a few things that you should look at and make sure you identify, and we've discussed it in the other video as well. But here's a membrane, like any other membrane. This one happens to be the inner mitochondrial membrane. And here's the inner membrane space. So here's the inner mitochondrial membrane. There's another membrane out here that's to the outside. Over here is a cell. There's the cytoplasm out here. So there's this space here that fills up with hydrogen ions, almost like a battery. And then over here is a mitochondrial matrix where things happen, right? So these proteins here are acting like that sodium potassium pump, okay? These proteins here, what they're doing is allowing the hydrogen to come in, but then they don't let it come out. So the hydrogen has a one-way ticket. These hydrogens go in this direction, but they don't get to come out. So remember, this is a key issue, because what we want to have this high keep high. We want to maintain this high concentration, and we want to maintain this low concentration. So the way we maintain that is we have to keep drawing these hydrogens in. Well, how do we draw the hydrogens in? We need something. If these are positive, we need an opposite, don't we? We need a negative. We need a negative. And what would that negative be? It's an electron. And where are the electrons coming from? They're here. There's these little stars. Those are what represent the electrons. And what is carrying those electrons? That's that NADH and FADH2, right? But the point of why I bring it up here, membrane transport, is I think that this is what confused some of you. When you see these hydrogen ions passing through the membrane, you think that they pass through the membrane. They're not passing through the membrane. They're passing through these carriers that require energy to do it. This is active transport. So remember, this is definitely active transport. And because it's active transport, you need energy to do it. You're not getting in the form of ATP. You're not using ATP, right? But it is another form of energy called electrons, electricity, basically, that's pulling the hydrogens up, and this movement of electrical charge or current is allowing then the hydrogen to be moving from this low concentration into the high concentration, and where it's trapped physically because it can't get back out through these carriers. And each of these carriers have names, and you have this intermembrane carrier called quinone and cytochrome C and different cytochromes that move back and forth and carry the electrons down. And each time the electrons move, it pulls in hydrogen into this intermembrane space where they're trapped. Now over here is another protein. Isn't that interesting? These are active transport. So this part here, this is active transport. So let's not get that confused. There's two kinds of transports happening here. All right. So this one's active transport, but this other one is different. This movement here is not active. This is passive. This is going from high to low. High to low. That's the way it's. You don't need energy for that to happen. These hydrogen ions will gladly go through this protein in order to let the hydrogen, allow them to go from high to low, easily go through this protein. This protein is known as ATP synthase. And the interesting thing is that as these hydrogens flow through here really quickly, they transfer some of their energy to this enzyme, which then turns it. It actually turns, and as it turns, ADP and phosphate. There's phosphate PO4 and ADP with two adenosine with two phosphates, ADP, and come together because of this enzyme, and you form ATP. ATP is being formed, not used. So don't get that confused. And I think this is what confused a lot of you during the test. I think you're kind of letting this example of both kinds of transport across a membrane. There are two different kinds of transport across a membrane, and they're being used in the mitochondria to create concentration gradient here, and then they're using the concentration gradient over here to produce ATP. The movement across the membrane here is passive in the sense that you don't require energy to make them move in this direction. Where over here, the movement across the membrane is active. So in this case, you're not using ATP, you're using electrons, but it's still energy, and you need to make sure you correct that in your mind, that this is movement of ions across the membrane, so that much is true, but you do have helpers, and that's what we said in the beginning. Ions do not cross the membrane without helping. So don't confuse that. Don't confuse that on a test when you're trying to answer questions. All right. So that's pretty much it. And what I wanted to mention with cellular respiration is where we're going to end up going next. So in cellular respiration, what have we done? Let's think about it. What have we done with cellular respiration up to this point? And make sure you've done it if you haven't done it. So in cellular respiration, the first thing we did is we talked about glycolysis. All right, so what was the point of glycolysis, and where did it, you have to know where it happened, where it happens. You have to know what does it do. And then you have to know what does it make? What does it produce? It produces what? Do you have to know all the steps? No, but you have to know that and you're doing that project now. You've gotten the lecture notes, you've gotten the video, you've gotten the you have the project, the activity you're doing. So you have to know where glycolysis happens. It happens in the cytoplasm. What does it do? It breaks glucose up into two pyruvate molecules. It produces two pyruvate molecules. It produces a net of 2 ATP, and it produces NADH. So those are the three big products of glycolysis. And what's the next step? Well, the next step is it can be broken up into one or two spots, right? Either one way or the other. If there's oxygen, it goes this way. And remember, oxygen passes through the membrane easily. It's oxygen gas, right? Or it doesn't pass because there's no oxygen. So no oxygen gas. It goes this way. In this direction, it's called, what is it? You tell me. Hope you said fermentation. And fermentation is important to society and to your life as you're running 2 miles on track. Fermentation, there's two kinds, lactic acid, and there's ethanol, right? And those are two kinds of fermentation, ethanol or lactic acid. And what you got to understand when you're dealing with lactic acid or ethanol fermentation is that those two things these are not healthy. We don't want them. And that's part of your project, is to figure out why we do it. And I think I've answered it in the video, actually. So if you pay attention to the video, the previous video, you should be able to answer it. The next step that you have to go, if you do have oxygen, so that's fermentation. That's what happens when you don't have oxygen present. But if you do have oxygen present, we do aerobic respiration, and in aerobic respiration, the next step is to go to the mitochondria. And in the mitochondria, in mito, there's two processes, right? The first one is the cycle. The TCA, there's three names for it: tricarboxylic acid cycle, TCA cycle, it's the first time I've mentioned it, but it's in the book. The other two names we've discussed in class, which is the Krebs cycle, right? Or the other name for it is you tell me. Okay, I hope someone said citric acid cycle, right? And so those are the three names, and here there's all kinds of things. The main purpose of this is to produce more electron carriers, and what happens here? Where does it happen? In the mitochondria. What does it do? It breaks up the pyruvates into carbon dioxide, and it takes the electrons and sends them down the line. What does it produce? A lot of NADH, a lot of some FADH2, and some GTP or ATP. And so what does it happens then? Is the next step? What's the next step after that? Is the electron transport chain. Where does that happen? In the membrane of the mitochondria, in the inner membrane of the mitochondria. What does it do? Electron transport chain uses a chemiosmotic gradient and it uses some active transport by using the electrons to pull the hydrogens in, creating a concentration gradient, and it uses ATP synthase to make ATP from ADP. And this is where oxygen is used, right? This is where oxygen is the last electron acceptor. All right, so that's it. That's what we're at. And I hope you're doing that project. I hope the project goes well tomorrow. Everybody's happy. I hope this helps to some extent mediate some of the issues that we had in that last quiz. It was really disappointing to see the quiz turn out the way it did. So we need to work a little harder. We'll think of something. We gotta think of something on how we can get this stuff in our heads quicker, or at least more efficiently. And come to tutoring if you need to, but we need to get this through.